My name is Matt Hipsey. I'm just here to introduce our speaker for this afternoon's session. Our speaker is Mike Venarsky. He's snuck into WA just before we've closed the border. It's been here since just March this year in a new role at DBCA, and we're going to hear a bit about that later. So Mike's come from the US. He's got his PhD from Alabama before in aquatic ecology. Before coming over to Australia, I'm working at the Australian Rivers Institute for a few years over in Queensland, and I think we're very fortunate to have got him over to the West. So he's going to be doing some starting new research and monitoring program of wetlands in Western Australia, continuing on some of the early work from Jim Lane and others, going back to the 1970s, monitoring how these wetland ecosystems have been changing. So I think we're going to talk in two parts from Mike today. First, a bit about his background, but also learning a little bit about what he's going to do going forward as part of his new role here at the DBCA. And we're sort of very happy to have the opportunity to hear about your ideas and get some input and opportunities for collaboration in this very important topic.
Thanks everyone for tuning in. Yeah, as Matt had mentioned, I've split this presentation up into two pieces. About half of it is my background, just to kind of give you a feel for who I am, the projects that I've been involved with over the years, and hopefully that'll give you a little bit of insight whenever I start actually talking about the Ramsar research and monitoring program that we're putting together. One aspect of the research and monitoring program that we're putting together that I'd like to stress with you, we're in the early stages of this at this moment. So consider this an open discussion. So comments, criticisms are welcome. The better that I understand where my blind spots are, the better that we'll be able to put this program together and hopefully make it successful. So with that, let's get stuck into it. Let me see here. There we go. Yep. Okay. So as you can probably tell, I'm not an Australian. I'm originally from the US. I grew up in the Midwest in the state of Illinois. And to put a picture in your mind's eye, think of scorched earth agriculture and corn and soybean fields for as far as the eye can see, right? I did my undergrad and master's at Southern Illinois University, Carbondale. And the projects that I was associated with at that time were much more on the applied end of the spectrum, focusing on conservation of threatened and endangered species and cave ecosystems. We did studies like life history studies, looking at growth rates and time to maturity, but also some very basic and at this time quite dated genetic studies looking at phylogeographic relationships between different populations of cave species. Now, after SIU, I moved to Tuscaloosa, Alabama, and started a PhD at the University of Alabama. And at that point, the projects that I was involved with shifted more from the applied end of the spectrum to much more basic ecology. And most of the questions, or most of the projects that I put together during my PhD were centered around answering this question or trying to figure out how energy availability shapes cave stream communities. And we use various techniques in this, food web studies that used combinations of gut content analyses and stable isotope analyses. And we also conducted a stream reach scale litter amendment experiment. And so within that particular experiment, we tracked the response of an entire cave community from microbes all the way to top predators, right? So after Tuscaloosa, I moved to Fort Collins, Colorado, and this was, I don't know, the period of time that I like to describe it as coming out of the dark and into the light, if you will. So I no longer worked in cave systems. I started working in mountain stream ecosystems. And this was one of these projects that was, kind of truly multidisciplinary. This was my first experience in this kind of group where we had biologists, biogeochemists, landscape level modelers, and fluviogeomorphologists. And most of our projects were centered around trying to understand how landscape disturbance history shapes stream and riparian ecosystems in the Mountain West. And so For our portions of the study, we focused on variables such as stream invertebrates, trout, emerging aquatic insects, and riparian spiders. But one aspect that I'd like you to keep in your mind as I describe this study is how important it is to scale your results to the appropriate spatial scale. And that's one of the important lessons that I learned out of this study. So let me walk you through some of the results for this. Or actually, first, let me give you a bit of background on these systems. So these mountain streams flow through these alternating unconfined and confined river valleys. And it's really in these unconfined valleys that the stream can take different forms. If you have a secondary growth or young growth forest, a forest that's either been hit by a stand-killing wildfire or has been logged in the past, the streams and rivers in those unconfined valleys typically are single channels, right? And on top of it, these are mountain systems. So these are high gradient systems. They're kind of like a fire hose shooting down the mountain. And there's a lot of erosional habitats and large substrates in these streams. There's not very many depositional habitats. So it's a fairly homogenized, if you will, right? Now, if you happen to be in an unconfined valley that has true old growth forests, well, in those systems, you have big trees. Those big trees fall into the river. They create log jams. Those log jams slow the water down. They bring the groundwater table up, and they allow multiple stream channels to form, but also allow a lot of depositional habitats to form. So what we have is we have streams in different forms, right? Or at least different shapes, if you will. And so we're trying to understand how those different shapes or those different forms of those streams influence the ecosystem processes in these mountain systems. Now, I'm not going to go into each of all the details of our results because I think just looking at one response variable will give you an idea of kind of the core findings that came out of this study. So here I have invertebrate secondary production. So this is an actual measure of the turnover rate or the production rate of animal biomass in these systems over time. And the first graph I have here is at a per unit area scale, all right? So this is commonly what you'll see ecologists report in the literature, right? We go out to these streams, we put our core in or we put our Hess sampler in, we dig all the bugs out, do our IDs, and then scale all of our estimates to a per square meter scale. And so what we see here is we have our old growth forests. So those are the streams that are in that multi-channel state. And then we have our young growth forest, those streams in that single channel state. And whenever we look at secondary production among these different stream forms, we don't really see much of a difference. There's no consistent difference between them, right? But once we rescale these numbers to the what we call the per unit of stream valley scale, we see a much different picture. And what we did here isn't complex, right? It's just multiplication and division. You just add up the number of square meters of pool habitat and riffle habitat and wood habitat, and you divide through by the length of the stream valley. And you can see by rescaling those numbers how much of a different picture we get in our results. In particular, we have one of our study sites that is just much, much more productive than all of our other study sites. And that's quite simply just driven by the amount of stream that's in that valley, right? It has 4 times more stream area in that valley compared to all these other study sites. Now you will note here that one of our old growth sites wasn't much more productive, or wasn't any more productive actually than our young growth study sites. Now, this site still had multiple stream channels. It's just that those stream channels weren't as big. And so it didn't increase the amount of stream area in that valley as much as it did in this other old growth site. But one thing I'd like you to note is the distribution of productivity in this old growth study site. You'll see that we have a decent amount in pools, riffles, and wood habitat, whereas in our young growth study sites, it's pretty much all riffle habitat. So now think about this from the perspective of trying to understand how these systems are responding to landscape disturbances. If you went into this system and just collected your samples and just expressed your results at the per unit area scale, you would see no difference in these study sites and may conclude that, well, for the most part, these streams and rivers have recovered from either logging or stand-killing wildfires. But it's only once you scale those estimates to the stream valley that you start to see the effect of those landscape disturbances, those historic effects. And so that can just be as simply as just the shape of the stream or the amount of wetted area in the stream. And if we think about how humans affect stream and river systems, among the primary ways that we affect them is by modifying the geomorphic processes that influence stream shape. So that'd be sediment dynamics and discharge regimes, right? And one other thing to note, I forgot to mention this, but the communities in these streams are all pretty much the same. There's no difference. So any differences that we're seeing here in this per unit of stream valley or any of the differences in the per unit area estimates are not due to differences in community composition. So just a few take-home messages. I've already kind of rammed home at this point. Stream form strongly influences ecological function, but the influence of stream form is dependent on the scale at which you express your variables or your results. Okay, so from Colorado, I moved to Brisbane to start a position with the Australian Rivers Institute at Griffith University. And during this period of time was when my projects shifted from that realm of basic ecology and really moved back into the realm of applied ecology. And the projects that I'm still associated with out at Griffith, because we're still working through some of those analyses, are focused on trying to understand how water development will influence both freshwater fisheries and estuary productivity in relation to migratory shorebirds. Now, for the purposes of today's talk, I'm just going to focus on our project in the Mitchell River here. Essentially, the Commonwealth would like to install dams at various portions in the headwaters of the Mitchell River. And so what we did was develop a project that looks at fish migration throughout the Mitchell River to try to understand how those dams may influence fish movement. Excuse me. So now, one thing to know, so connectivity is this central tenet in ecology. And one way that we can measure connectivity both within an ecosystem or between ecosystems is by looking at movement patterns of species. But one of the things that's kind of stood out to me in the literature is that, well, first off, many species movement studies focus on charismatic megafauna or commercially or recreationally important species, right? They don't typically look at community level movement patterns. Now, in the fisheries literature, there are some studies that look at community level movement patterns of fish, but they tend to be at relatively small spatial scales. So you might be looking at fish movement patterns over a river segment that might be, let's say, several hundred meters up to maybe a couple of kilometers long. But they very rarely, or they are not, at least to my knowledge, done at the scale of the entire river catchment. And that's what we wanted to do with this particular study. First, we wanted to focus on, or try to understand, the movement patterns of the entire, well, entire in quotes there, as many fish species throughout the Mitchell River as possible. So from little gobies all the way up to big barramundi that are over 1 meter long. And we also wanted to look at these movement patterns of fish in different portions of the catchment. So we have sites that are in the headwaters and in the midreaches and in the floodplain, right? Because if you're in different portions of the catchment, that might influence how these fish move through the catchment. So we did that by using otolith microchemistry analyses to track fish movement. And so I'll run through this, this approach real quick, just in case people aren't familiar. So fish have these little bony structures in their head called otoliths. And as the fish grows, the otolith grows along with it. So what we do is we capture a fish, we sacrifice it, remove the otolith, and then we do a cross section through that otolith. And that's what this is down here on the lower left-hand portion of the slide. This is a Barramundi Otolith. And if you look real closely, you might be able to see rings in there. You can actually count those numbers of rings and get an estimate of the age of that particular individual. So once we have these cross sections, we then set up a laser ablation transect that starts at the core, which is age 0, so that's when that fish was born, and runs to the edge, which is the time that we sacrificed that individual for these analyses. And as that laser is running across that otolith, it's measuring the concentration of strontium. But more specifically, it's measuring the concentration of 2 stable isotopes of strontium, strontium-87 and strontium-86, okay? So we look at the variability of strontium across that otolith, and then we also map out the variability of strontium throughout the river catchment by analyzing water samples for the same strontium isotope ratio, right? So then we compare that transect and the otolith to the spatial variability, and we can get an idea of where that fish might have spent time at different portions of its life, okay? So in total, we collected fish and water from 25 sites throughout the Mitchell, totaling 987 otoliths across 27 different species. Now, one other aspect of this method that we need to account for is temporal variability in strontium. So the Mitchell River is a wet-dry tropical river. So it basically fluctuates from a flowing river during the wet season to basically a chain of lakes during the dry season, or a chain of disconnected water holes. And those changes in discharge can have a strong influence on the strontium isotope composition. So mussels are sedentary, or at least relatively sedentary. So we collected mussels, cross-sectioned those mussel shells, and did the same laser ablation analyses on them, right? Because that way we can use mussels to get an idea of what the variability is in strontium at any particular site throughout the Mitchell River catchment, right? So we're accounting for spatial and temporal variability in these analyses. Now, I think now finally have got the model done and have just worked through our first full set of analyses of this data set. So what I'm presenting here are some preliminary analyses, but I don't expect these results to change too much with the new batch of results that we have. So the first thing I'll describe is the spatial variability in strontium across the Mitchell River catchment. So on the y-axis here, we have that strontium isotope ratio, and on the x-axis, we have distance to river mouth. So that's just to kind of give everybody an understanding of where these sites are located in space. The red circles are the Palmer River, so they sit up here. We've got the blue squares, the Mitchell River in the middle, and then down on the bottom here, we have the Walsh River. And what this is indicating to us is we've got a fair amount of separation between our subcatchments. Over here, we have the Palmer, the Mitchell, and the Walsh. So we've got enough separation in those subcatchments to, at the very least, track the movement of fish from, let's say, the Palmer River to the Mitchell or vice versa. Now, we also have an updated version of this isoscape, which I'm not going to present today, but we also have the ability in certain portions of the catchment to look at upstream and downstream movements as well. So we've got a decent amount of resolution using this particular method. This method of tracking fish movement is not to track fish movement over small spatial scales, it's to track fish movement over large spatial scales. Over the entire catchment is really the power. So, okay. All right, so here we have the white triangles are the average strontium isotope ratio from the mussel shells, and those bars are the variability. So what this is indicating to us is that, well, it appears, at least the variability that we're seeing within these mussel shells is not larger than the spatial variability that we're seeing between our subcatchments. So this should suggest to us that temporal variability in strontium is not influencing our results all that much. So now I'm just going to ask a couple of very simple questions to give you a feel for what some of these, what some of our results are looking like. I haven't drilled into species-specific patterns yet, so these are just for the entire fish community. So the first question is, were you born at the site of capture? Where we collected you, were you born there or did you come from somewhere else? And I broke this into two different types of habitats, main channel habitats and then off-channel water holes. And the reason for that is they have different hydrologies, right? Those main channel habitats connect quite a bit more often, especially during the wet seasons, even during small wet seasons. But those off-channel water holes need certain conditions for them to either connect with one another or back to that main channel habitat. And so what we're seeing here is that, well, only about 32% of the individuals that we collected a main channel habitat are from that region from which we collected them in. So it appears to be fish are moving around quite a bit in your main channels, which makes sense. But in our water holes, it's only 79, or excuse me, as there are 79% of the individuals are from that water hole, right? And again, that makes sense because these are more isolated portions of the catchment that don't necessarily connect up with either main channel habitats or other water holes throughout the year. And then if we split those numbers down by subcatchment for the main channel sites only, I mean, there's a bit of difference here between our different subcatchments. It seems to be fish are moving around a bit more in the Palmer River than they are in the Walsh or the Mitchell. But right now, you know, I wouldn't put too much confidence in those estimates because we do still have to get errors estimates around those. Okay, so now another question that we can ask with this data set is a simple one. It's just like, how many times did you move after you were born, right? And what we see is 35% of these fish don't move at all. Another 30% only move one time, and then the remaining 25% move 2 or more times. And these results align pretty well with how we understand fish movement from other species in other parts of the world, meaning that the majority of fish at a particular location appear to either not move very far or not move at all over their lifetime. You have a very small portion of your population that are your big movers, they're your colonizers. And arguably, they're among your most important portions of the community, right? Because if you have a disturbance event that takes out a water hole, or maybe a water hole dries up completely, well, these are your individuals that are going to recolonize that habitat, right? They're your big movers. They're out there seeking new places for them to establish themselves. Okay, so where are we going with this in the future? I'll just highlight a few aspects of the way that we conducted this study to kind of give you an idea of where I think we can take this data set. First off, we did not conduct our sampling by just going out to a site and collecting representative fish from all of these different species. We actually did this to quantify community composition, more specifically relative abundance and relative biomass of different fish species at each of these study sites. So we're going to be able to weight our movement data by relative abundance and relative biomass. And that can begin to give us an idea of, well, you know, a coarse idea of the numbers of fish moving through this catchment and the biomass of fish that are moving through this catchment, okay? On top of it, the method that I talked about today is just strontium, but we also collected bulk stable isotope samples from muscle tissue from all of these individuals as well. And the reason for that is that this is stable isotope analyses, right? So when it works, it's great. And when it doesn't, it's like trying to read tea leaves, right? And it can be a very, very frustrating process. So what we're hoping to do with this data set is, well, we can build an isoscape with strontium, but then we can also build an isoscape with sulfur, carbon, and nitrogen and kind of lay these stable isotopes over the top of one another. And then if maybe we can use other stable isotopes to get an idea of movement patterns if one of them isn't working too well, right? So that's something that'll be kind of in the latter stages of our analyses that we're going to start to try to put together. And you can also put other variables, environmental tracers in here as well, such as metals if you wanted to. Okay, so enough about what stuff I've done in the past. Let's move on to the Ramsar Research and Monitoring Program. So as I mentioned before, this program is still in the development phase. So please, questions, comments, criticisms about the way that we're approaching this, all of it's fair game. The more that I can interact with people and pick their brains on these issues, the better, okay? Now, just to give everybody a quick rundown of what Ramsar is. The Ramsar Convention, I think, was started back in the 70s, if I remember right. And it's changed a little bit over the years, but basically it's there for wetland conservation. Its aim is to preserve unique wetlands, whether that be a unique hydrology or a unique biology, and also preserve wetlands that are important waterbird habitat, either feeding habitat or roosting or nesting habitats. Globally, there's more than 1,800 wetlands that have Ramsar designation. 66 are in Australia and 12 are located in WA. And one thing to note about these sites is a lot of them are wetland complexes. So we have over 100 wetlands that are included in our Ramsar sites across the state. And the organization that I now work for, DBCA, is responsible for reporting on wetland condition. Now, the design and implementation of this program has to overcome several issues. And so what I'm going to do is I'm going to highlight some of those issues, and then I'm going to give you an idea of how we're approaching those issues. And I think that'll, well, again, we're early on in this process, right? So it's still under development. Now, one of the first factors we've got to deal with is there's a lot of cooks in the kitchen at these study sites. DBCA is doing some monitoring. The Department of Water and Environmental Regulation is doing things. Natural resource management groups, local communities, and universities. And I imagine maybe even some of you watching this have conducted a study or two, an ad hoc study or two at some of these wetlands. And that presents an issue for us, right? Because we have some monitoring going on at most sites, right? It's not necessarily all done the same way. Plus, we have data and information that's scattered amongst a multitude of different agencies. Hell, some of you may even have data sitting on your computer right now from some of these study sites. So our first step in this process is what we're calling building the map, or trying to collate all of the existing information, or as much of this information as possible from all of these Ramsar sites, into a centralized bibliography and or data repository, right? So this will help us get an idea of what's been done and what is being done at all of these sites so that we don't duplicate efforts. And this will also help from a reporting standpoint, right? It's best for us at DBCA to have this information in a single place so that whenever we do have to report on it, well, we know that we have the most up-to-date information. And as well, for our collaborators in the future, if somebody is interested in, say, putting a project together for Forestdale Lake, well, we can hand over the data repository that's for Forestdale Lake, and then that person has at least most of the information associated with that particular study site. So we not only see this as a use for internal at DBCA, but also with our collaborators and anyone else that might be interested in that, for that matter. Okay, a couple of other issues that we've got to overcome. Every site is unique, right? We've got sites up outside of Broome, Roebuck Bay, and 80 Mile Beach that are quite a bit different than, let's say, Forestdale or Thompson's Lake here in the Perth metropolitan area. So how we monitor these sites, or the variables that we collect at these sites, is going to vary from place to place. Additionally, so limits of acceptable change. So there can be an issue surrounding these limits. So let me describe what limits of acceptable change are first. For each of these study sites, for each of these Ramsar sites, they put together an ecological character description. And basically, it's just a review paper of the site. So it summarizes all the previous research and lists the criteria of why the site got Ramsar designation. But also at the back of these, they provide limits of acceptable change around various environmental variables. And these limits are, well, let's walk through one. For example, let's say they have a limit of acceptable change around water depth, saying that the water depth of a particular wetland varies from 0.5 meters to 2 meters over the course of a year. Well, if we start to see water levels outside of that range, that triggers actions by us, right? So we may go out there to figure out what is causing this increase in water levels, and then determine whether or not those increases in water levels are actually having a negative impact on the study site, right? So that's how we use these limits of acceptable change. It gives us an idea of if these systems are changing. But the issue with this is that the limits of acceptable change were often developed with the best data available at the time, not necessarily the best data to place bounds around those limits of acceptable change. So another aspect of this monitoring program is filling in the gaps, right? Developing site-specific monitoring and research projects to address knowledge gaps, whether they be around limits of acceptable change or trying to figure out what the best variables are to monitor at this study site so that we can keep an eye on its condition. And I see this as being one area, well, very fruitful area for collaboration, right? And we'll be working quite a bit with regional managers as well, because they are the people out there most often. So they have a good idea of what information they need to know about these sites in terms of their management. All right, and then the other issue that we've got to deal with is, and this is always just kind of the elephant in the room with monitoring studies, we can't monitor everything, right? So what we're thinking about doing with this particular aspect of it, right? Well, we can't monitor everything because, well, first off, we don't have infinite amounts of money, and it's just me that's associated with this program. So what we're thinking about doing is attempting to construct a common foundation in this monitoring program and try to focus on a habitat template approach. And I'll walk you through what I mean with this habitat template approach, but just from a kind of a theoretical standpoint, if you will, habitat controls everything about ecosystems. At least, I don't want to say everything, but most things about ecosystems, right? It determines what species are there. It'll determine how long species are there. It'll determine how many individuals within a species are there, right? So it has a lot of influence on contemporary ecological processes. But the habitat template also influences evolution, right? Because as habitats change, species can change along with them, whether that be life history or morphology. So this habitat template concept is this kind of core concept that's built both in our understanding of streams and rivers and also terrestrial environments. So let me walk you through what we're doing with a long-term data set that I came across at DBCA to hopefully illustrate how I think we can utilize this habitat template approach to monitor Ramsar sites across the state. So let me give you a little bit of background. I'm sure some of you are aware of this data set, but my predecessor, Jim Lane, conducted a long-term and large-scale monitoring program of wetlands across the Southwest, and it's called the Southwest Wetland Monitoring Program, or SWAMP for short. And this just gives you, it's a big scale from Esperance all the way to Perth, right, in this red area right here on the map. He monitored water depth and basic water chemistry at approximately 167 wetlands from 1977 to 2014. And this is one of these data sets that is absolutely, without a doubt, second to none, right? And it has yet to be analyzed in its full extent. It's been picked apart, right? Meaning that people will work on individual wetlands and they'll take information from this data set, but it has not been analyzed in totality, right? On top of it, on top of just the water chemistry and the water depth information, for a 15-year period at 15 wetlands, they monitored vegetation, aquatic inverts, and birds. Right? So it's already a great data set, and then we're adding another great data set on top of it. Okay? But like with any data set, it's not perfect. Right? Sites were only visited twice per year. Some sites were visited for a few years and then skipped for a few years, and then people would go back to them for a few years. So there's a lot of gaps in the data set. And while we can't fill in the gaps in water chemistry, we can fill in those gaps on water depth. And as I walk you through the process of how we're going to try to do that, hopefully that'll illuminate this habitat template approach a little bit better in terms of what I think we can do at some of these Ramsar sites. And one thing to note, some of the Ramsar sites were part of Jim's wetland monitoring program. Okay, so here in the upper left-hand corner, I just have this simple graph of water depth on the y-axis and then time on the x-axis, and you can see all these little just individual points for whenever Jim collected a water depth measure. So what we do is we take Jim's measurements, and then we go back into the Landsat archive, and we pull out a Landsat image of that wetland that's relatively close to whenever Jim went out there to measure water depth. We then can estimate water surface area from that Landsat image, and then come up with a relationship that relates water surface area and water depth. Now, the relationships are not going to be this nice, and it's going to have a lot to depend on the geomorphology of the wetland, right? If you have a shallow clay pan, decreases in water depth will probably correlate relatively strongly with decreases with decreases in water surface area. But if you have a wetland that's more like a bowl, right, with steep sides, decreases in water depth are not going to correlate all that well with changes in water surface area. All right? So we'll figure out how many wetlands this is actually going to work with. So once we have this relationship, we can go back into those archived Landsat images, which go back all the way to the 80s, and we can then basically come up with what I'm calling just a gap-filled water depth profile for these wetlands. And of course, it's not going to end up being a continuous water profile. We'll still have gaps in there, but it will be a less gappy data set, if you will, right? So just with this data set alone, some of the questions that we can answer, we can characterize the wetlands based on their hydroperiod. Given that this data set goes so far back into history, we can begin to get an idea of the environmental variables that are driving changes in hydroperiod. May even be able to develop predictive models to try to understand how these wetlands may change in the future. And then also with that vegetation, bug, and bird data set that we have, well, we can begin to look at changes in those aspects of the community in relation to hydrology and see if there are any eco-hydrological archetypes. So, but we can still take this data set a bit further if we add some more information onto it. So if we take this gap-filled data set, and then if we're able to begin to build a database of bathymetry of these different wetlands, and maybe even get some detailed maps of vegetation distribution, well, once we combine all of this data together, we can start estimating water volumes of these wetlands, which are core components of hydrologic models. right? Even coarse hydrologic models can use these water volume estimates, right? But we can also begin to then quantify aspects of habitat in relation to various species. And one of those could be water bird feeding habitat, right? With this bathymetry and this water depth, bringing those two metrics together, we can get an idea of, well, how deep is that water? Not only how deep that water is, but how much of that water is there within a certain depth, right? So waterfowl like to have water that's deeper than a half meter, and waders like to have water that's less than a half meter deep. So how do the proportion or the amounts of those habitats change in space and time? And of course, if we have these vegetation distribution maps, we can look at time of inundation of different vegetation units. But then one of the ultimate goals with something like this is to link aspects of habitat. I'm using vegetation distribution in this example, but can we link those aspects of habitat to remote sensing images, right? And then allow spatial and temporal changes in vegetation to be tracked remotely. Because some of these study sites are pretty remote, and even some of the regional folks only get out there a couple of times a year. So what we're trying to do is use a combination of on-the-ground techniques and remote sensing techniques. And we're hoping to fill in that with maybe some drone mapping of vegetation using drones, right? So that rather than just looking at pixels from remote sensing images, can we begin to get a better understanding of what's in those individual pixels? So hopefully that gives you a bit of a better idea in terms of how we're approaching this using this habitat template approach within this Ramsar research and monitoring program. And with that, I'll take any questions or comments people might have.
Well, great. I think with that then, I'll say thank you for coming along and sharing your past work, but also the opportunity to have some feedback into your new work. And I'm sure if people have further questions, you can contact Mike on his email, which I'll put up on the screen after this. Yep. So, and yeah, thanks a lot for coming down. We really look forward to seeing the re-emergence of the swamp monitoring program.
Yes. All right. Well, thanks for having me. I appreciate everybody's time.
Great. Thank you.